Hello everyone, my name is Rebekah. I'm a fourth year animal biology student, and this summer I worked as a USRA student under Professor Alexandra Harlander. The main task I was involved in was artificially inseminating 3 lines of hens genetically selected for feather pecking behavior. And having never touched a chicken before this summer, it's safe to say that I learned a lot and I'm excited to share some of that with you today. Our lab spent a lot of money a number of years ago bringing these 3 lines of birds to Canada from Europe. Since they were rather expensive to bring over, it is important that we breed them every year so that they are available to graduate students who may want to study them. The 3 lines have known genetic differences that contribute to their behavior. We have the low feather peckers, the high feather peckers, and the control groups. Our breeding program consisted of 38 roosters and 114 hens. We select for feather pecking behavior not only to study the behavior itself, but also to have increased differences in the physiological pathways linked to the behavior. So here's an overview of the whole process that I will be going into detail about. The basic idea for the selection process is to breed birds with no ancestors in common on 2 generations to avoid inbreeding. We then breed extreme phenotypes together, the highest feather-pecking males with the highest feather-pecking females, for example. We keep a detailed Excel sheet where we record the relations of each generation of birds. We have established families inside each line of birds in order to keep inbreeding to a minimum, because too much inbreeding can negatively impact the fitness and reproductive capabilities of the birds. So the first step is semen collection, which is called milking the rooster, and there are 2 roles involved. The first is a stimulator who holds the bird in place and strokes up the back to stimulate semen release. Then the collector brings the tube to the cloaca to collect the semen. The picture on the right is an example of a good collection sample, and what I mean by this is that it's a good quantity, it's white and opaque rather than clear and watery, and so this is ideally what you want to get from every rooster. The next step is to inseminate the hens. We organize our collection tubes on this colorful table that clearly outlines which rooster is inseminating which 3 hens. It is important at this step to be organized to prevent any mistakes from occurring. We did this whole process for all 114 hens twice a week because chickens actually have a way of storing the semen and can produce multiple fertile eggs off one insemination. During the 4 to 5 weeks of breeding, we had to collect the eggs from the hens every single day. Each egg would be labeled with the date, family ID, and hen number so we can keep track of which egg came from which hen. We store the eggs in a cooler at 15.5 degrees Celsius until we are ready to move them to the incubator. Also, just as a disclaimer, our lab values animal welfare above all else, and our birds were only kept in individual cages for the duration of the breeding program before being moved back to their floor pen enriched group housing that you saw on the last slide. But there is no other way to be able to track where each egg is coming from without this temporary housing system, and we try to make it as positive an experience as possible by giving them lots of corn after each insemination. Once we got enough eggs, we had to sort all the eggs by line and by hen and load them into the incubator. The picture on the left are just the eggs I sorted for the high feather pecking line, and the picture on the right is all our eggs loaded up, and it's hard to tell from the picture, but there were over 1,600 eggs in that incubator. The eggs will stay in there for 18 days at 37.5 degrees Celsius at 55% relative humidity and 24 turns a day to allow for even development. The time from incubation to hatching is 22 days, but the last 3 to 4 days are spent in the hatcher. The next step was to candle the eggs. So, as you can see on the left, an unfertile egg will just have a yolk that you can clearly see through, but fertile eggs will be mostly opaque with blood vessels. We moved all the fertile eggs to the hatcher where they will stay for 3 to 4 days at 37.5 degrees Celsius and 90% relative humidity. On the right are just some pictures of the loading process and the hatcher itself. So after 5 weeks of inseminating and 3 weeks of incubation, we finally reached hatch day, which was the best day of the process. It was so rewarding seeing all the baby chicks. We started the day by sorting all the full sibling chicks. My role on hatch day was to assign and record the wing tag number to all the chicks. We use this as a source of identification, and it just goes in the skin of the wing. The red tags are the low feather peckers, the green tags are the high feather peckers, and the yellow tags are the controls. The next step was to vaccinate the chicks. They receive one injectable vaccine and one spray-on vaccine, which is green in color and why the chicks on the right look wet and slightly green. So here are my key takeaways from this summer. I worked with an amazing lab team who taught me so many new things. I was new to the poultry world, but I absolutely loved working with the chickens and learning about rearing, research, breeding, behavior, and more. I want to give a big thank you to everyone in the Harlander lab for a fantastic summer. It was a pleasure working with all of you. Thank you for taking the time to listen to my presentation. I hope you learned something new about chickens today. The final step was to move the chicks to their pens and get each chick used to the nipple drinking system. This hopefully increases the chance of survival because dehydration is very dangerous to baby chicks. Into their own baskets, counting them, bringing them to the sexer, because yes, sexing chicks is a highly specialized job. Then we selected the best chicks for wing